We continue with our course. We arrive to the second type of genetic mutations, the mutation by deletion. Here we have the DNA transcribed strand, a DNA transcribed strand, an example. We have represented three codons, ACG, TTC, AGT. So the corresponding mRNA, which is complementary to this DNA transcribed strand, is U complementary to A, G complementary to C, and C complementary to G. The second codon will be AAG, the third codon will be UCA. According to the genetic code table, UGC codes for cysteine. So UGC, UGC codes for cysteine. AAG codes for lysine, and UCA codes for serine. Now, what could be the consequence of the deletion of the second nucleotide in the second codon, T? When T is deleted, ACG, the first codon, does not change. However, in the second codon, C will be displaced backward to replace the deleted T. And A will replace C here, G will replace A, and T will replace G. We have a frame shift, a displacement of the reading frame of the DNA strand. So we obtain in the new DNA strand ACG, T, C, A, and not TTC. So T here is deleted, C will be displaced backward to replace T. So the second codon will be TCA, and the third codon will be formed of two nucleotides instead of three. This leads to what? This leads to a change in the mRNA sequence, ACG, UGC. Here the second codon becomes AGU instead of AAG, since A is complementary to T, G complementary to C, and here U complementary to A. And in the third codon, GT, we have CA, but we don't have a complete codon. This codon is truncated, is incomplete. And thus, the amino acid sequence will be formed of two amino acids instead of three. Why? Since the third codon cannot give an amino acid. Why? Because we know that one codon is made up of three nucleotides. Each three nucleotides give one amino acid. Two nucleotides cannot give an amino acid. Therefore, what could be the consequence of this deletion on the protein? The protein is abnormal, truncated. The protein is incomplete. It is constituted of two amino acids instead of three. Thus, a mutation by deletion is always, or it always causes a frame shift in the reading of the nucleotide sequence. It causes a displacement of the nucleotides and always affects the protein as well as the phenotype. The deletion mutation cannot be silent since it changes all the nucleotides that follow the site of mutation. As you can see here, all the nucleotides that follow the mutation site have changed. So the deletion is not silent. Now we pass to the third type of mutations, which is the mutation by insertion. It's the opposite of the mutation by deletion. Here we have a loss of a nucleotide. Here we have a gain or an addition of a nucleotide. So let's suppose or imagine that a nucleotide T is added in the second codon between A and C. So the consequence will be T is inserted into A between A and C. So now this nucleotide C will be displaced forward. T is inserted between A and C. Now we have a codon formed with A, T, C. The third C will pass to the next codon, and the next codon becomes CAG, and this T remains alone. The consequence will be the formation of three codons. The first codon is always the same. So the mutation by deletion and insertion doesn't affect the previous codons, but the following codons. ACC is complementary to AAG, while here ATC will be complementary to UAG, same as for CAG is complementary to GUC, and here we have only A phase 2T. As a consequence, according to the genetic code table, UGC will give cis, while UAG is a stop codon. Stop codon means that the amino acid sequence is stopped here. So the amino acid sequence is formed only of one amino acid instead of three. So the protein is surely abnormal and truncated. It's an incomplete protein. So same as for the deletion, the insertion could not be silent. It always affects the protein sequence, thus affects the phenotype. So, as a definition, the insertion mutation is the addition of one or more nucleotide that causes a frame shift in the reading of the nucleotide sequence, which means that causes a displacement of the following nucleotides, affecting thus the protein sequence and the phenotype. So the insertion is not silent, cannot be silent. It always affects the phenotype like the deletion, while the substitution can be silent. So here a diagram that summarizes all these types of genetic mutations. We have three types: substitution, one nucleotide is replaced by another; deletion, here G is deleted; or insertion, and A is inserted between T and G. The substitution can be silent, doesn't affect the protein sequence and the phenotype, or it can be missense. Missense means it leads to the formation of a protein which is non-functional. So missense, it misses its sense, its function. So the substitution may lead to the formation of a non-functional protein which affects the phenotype. Also, it can be silent. Also, the substitution can be nonsense, which means it leads to the appearance of an early stop codon, leading to the production of truncated protein, incomplete protein, which has no sense and surely affects the phenotype. So, missense mutation means mutation leading to the formation of a non-functional protein, while the nonsense means the formation of a truncated protein, incomplete protein that cannot function properly, leading to an abnormality in the phenotype. Now, the deletion and the insertion are never silent. They are either nonsense mutations or frame shift mutation. They may lead to the appearance of truncated protein or to an abnormally longer protein which is non-functional and which affects the phenotype. So you have to memorize this diagram. What are the three types of mutations and what are the consequences of these mutations? Substitution can be silent or missense or nonsense, while deletion, insertion are nonsense, and they cause a frame shift, a displacement in the DNA reading frame, leading to a truncated or abnormally longer protein. These mutations can be point mutations when they affect only one nucleotide, when one nucleotide is substituted or added or lost, or they can be stretched mutations when they affect many nucleotides, when two or more nucleotides at the same time are substituted or added or lost. So 100 and even thousands of nucleotides can be added, substituted, or lost. Also, these mutations can be somatic. They appear in the somatic cells, not in the gametes. And these mutations will not affect the offspring of the individual. For example, the mutations that occur in the skin cells due to the UV rays of the sun. In this case, we may have skin cancer, and this skin cancer is not transmitted to the offspring since the mutation doesn't occur in the gametes but in the skin cells of the individual. Also, the mutations can be germinal. They occur in the gametes or the reproductive cells. In this case, the mutation will be transmitted to the next generation due to the fertilization, the meeting of the ovum and the spermatozoa to give a zygote. This zygote will inherit all the characters, all the mutations from the parents. So somatic mutations are not transmitted to offspring, whereas germline or germinal mutations are transmitted to the offspring or to the next generation. In the part 2, we are going to solve some application exercises about these mutations. Thanks for listening.